Hello, BIM students. Welcome to the last day of Block 2. As you've seen throughout the week, we've been focusing on different transactions that are particularly focused on genomic architecture, DNA replication, its integration with the cell cycle, chromatin organization, and things along these lines. So today, what I will try to do is to cover an equally important topic that deals with chromosomal transactions that ensures the integrity of the genome. And so I divided this into two lectures. The first one will focus on different DNA repair mechanisms that are centered around fixing most of the commonly seen types of DNA damage that every given cell, whether dividing or not, are exposed to, and these include base excision repair, nucleotide excision repair, mismatch repair, and translation DNA synthesis. And then in the second lecture, we'll focus on repairing double-strand breaks, which are much more serious lesions that probably the most serious of all that genomic DNA can be encountered and must be repaired in order for the cell to maintain viability. And so we'll dedicate a whole lecture for that. All right, so let's begin. See how this works. All right, so I thought that reading is clearly important, and I realized that you probably have gone over the chapter in Albers, but I thought that including a few review articles can help you cope up with the topic. It's a very vast topic, and it's keep changing. As you can see, I included actually 3 different reviews from different timing. The latest, which is 2012. Since then, there have been a lot of more information that came up in this topic, and so it's a continuously evolving topic. But I thought that these are pretty good reviews, and if you're interested in the topic and maybe this is something you want to pursue later on, I thought that these are some of the best reviews on this vast topic. All right. All right, so here's an outline for the first lecture of today. We'll begin by surveying some of the major sources and types of DNA damage that a cell can encounter. We'll then summarize briefly what these types of DNA damages are, how can they be inflicted, and then the different repair systems that the cells have evolved to cope with these sort of lesions. We'll try to focus on those proteins that are relevant to mammals, even though a lot of what we know today about these DNA damages and repair actually been discovered in bacteria and later on in yeast, but the pathways are being pretty much concerned. Of course, mammals have developed a new set of proteins that are specialized to cope with certain types of DNA damage. And then I'll try to put this in the context of some of the diseases that are based on genetic perturbations, some of the genes that are required to repair these lesions. And then after that, what we'll do is we will sort of go over some of the fundamentals of these different repair pathways. I realize that this topic involves a lot of new terminology that you probably never heard of before. My task here is not to make sure that you memorize these proteins, but to understand the functional conservation of the different activities that are needed for these repair programs to be executed. And so that's my job. So don't worry too much if you've heard so many different proteins, but try to understand the principle of it. And I hope to make a good job at that. All right, so that's our outline for today. I think that always discussing some historical background is an informative thing to begin the topic. And so I'll begin with the discovery of a mutation, the term that was first coined in 1914, over 100 years now, by Hugo de Vries and his principle of theory of mutations, where basically they sort of propose the idea that genetic variation arises from unspecified intracellular perturbation. This is where the first time the co-mutation was sort of put forward. About 10 years after, maybe a little bit more, Hermann Muller have then proposed the idea of artificial transmutations of genes, and in his studies, he's been using Drosophila, the fly Drosophila, as an organism for detecting these different mutagenic effects using X-rays as a source of mutagenesis. This actually had a lot of impact on the field as a whole. As you can see from one single experiment, the number of different mutants of Drosophila that exhibited different various phenotypes have increased by 50%. All right, 1935, ultraviolet was used extensively as a way to induce mutagenesis in different organisms and bacteria primarily. And the concept of targeting a particular gene, of course, at that time, genes were known, but they were not known what they made of. But the idea that mutations are something that can actually affect genes was proposed at that time, 1935. Then Avery demonstrated that genes actually were made of DNA, basically nucleic acid that we know of today, the A, C, G, and T, right? These four fancy letters that you know of. We'll go over these, of course. You must have a good grasp of this from your undergrad studies, so we'll just keep going. All right, then later on, the concept of DNA repair have basically arise where Delbico and Kellner both separately discovered the idea that UV induction of mutagenesis can actually be counteracted by some sort of mysterious processes that different types of cells have. All right, and so that's where the idea that these different mutations can be inhibited to begin with, or if there's some damage to the DNA, actually can be counteracted by certain cellular phenotypes. All right, so we'll survey some of these different types of DNA damage. As I mentioned, DNA damage is a very ubiquitous term. It encompasses many different types of lesions in the genomic DNA. It can arise from normal cellular processes, just normal metabolism that induces a variety of different damages that we will just go over in a few seconds. Environmental agents are another source of these damages. Cells can also be exposed to additional, not necessarily normal environmental, but exogenous agent that can actually induce damage at a very heightened rate. For example, cigarette smoking is one such example. Radiation in space, this is another source of damage. Right. We also know that the accumulation of DNA damage is associated with developmental abnormalities, neurological disorders, cancer, cardiovascular diseases. These are all diseases that have some association with the accumulation of DNA damage in the cells. And this is where the importance of the topic comes into play. More specifically, in a given cell, in a day, there are more than 10 to the 5th DNA lesions every given cell is encountering. And that's not necessarily associated with dividing cells, as I mentioned. Even sitting cells, quiescent cells, cells that are already known to doing their normal function, they accumulate these spontaneous lesions, and they need to be corrected. You can imagine that, for example, a liver cell needs to transactivate certain liver genes to do their function, and these genes, in order for them to express the right proteins, they must express them from an intact liver-specific genes, right? And so if you have one cell that have damaged that particular specific gene, then that gene will not be able to function. Therefore, that gene must be restored to its normal architecture in order to be transcribed correctly and then translated into the functional protein. Of course, dividing cells are much more vulnerable to DNA damage, and this is where dividing cells accumulate a lot more DNA damage as a function of just normal errors that happen during normal DNA replication. Of course, the topic has been extensively covered, and we know that it's mediated primarily by these polymerizing enzyme or DNA polymerases that are have high fidelity, but nonetheless, they're not error-free. They can make errors, so maybe at a very low rate, but they still do make error, despite the fact that they do have proofreading function. And these errors need to be corrected as well. Reactive metabolites, such as reactive oxygen species, is another common source of DNA damage. And these are generated normally in the cells, but they're generated also at a much higher rate in response to different environmental agents, such as ionizing radiation or even sunlight. High energy methyl donors are another examples of common damaging agents, dietary nitrosamines, amines and bile acids, as well as chemicals in the environment. These are all sources of DNA damage. Of course, not included in this list is chemotherapeutic agents that we use for normal cancer therapy. They actually kill cancer cells because of their ability to induce DNA damage in the cell. And so that's another common source of extensive DNA damage. And you would hope that at that point, particularly for that particular purpose, that these damages are overwhelming enough so that the cells cannot recover from them. And that's the whole concept of chemotherapeutic efficacy. All right, so to the magnitude of DNA damage cannot be underestimated. And here's a summary table that I thought was very fascinating. This is how many different lesions are being repaired daily, right, in any given cell. So in any given cell, I'll just take an example. I'm not going to go through the entire list. You have it, so you can look at it at your convenience. But for example, just a normal hydrolytic reaction that happens in DNA, about 18,000 different events of depurination, removing the purines from the DNA structure. 18,000 event happens in every given cell daily, right? So, and these cannot go on answered. They must be repaired and repaired faithfully in order for them to happen. And so the cells must be able to deal with these kind of damages that are happening. And again, this is a reaction that doesn't require anything more than water. They do not depend on any enzymatic activity. They're not necessarily resulting from environmental exposure to certain environmental factors. So these are normal spontaneous reactions that happen all the time. Despite the stability of DNA, these DNA molecules are continuously generating damages that must be repaired daily. All right, so this is just to highlight one particular lesion. Of course, there are many different variety of lesions that we'll try to go over some of them, not all, but some of them, just to highlight the complexity of the topic. Of course, despite the efficiency of the cellular programs to cope with these lesions, some of them get fixed and they cannot be repaired. You would hope that these are the ones that do not interfere with the viability of the cells and do not result into a disease. And so from this vast amount, less than 0.2%, that's still a lot of damage that get fixed in any given cells in the genome. Of course, if these happen to be in gametes, then such errors will be inherited. It will be passed on to daughter cells and daughter embryos and individuals if we're talking about humans, right? So this is just why it's really important to repair these lesions. All right, here's a nice diagram that I found very telling that sort of summarizes all kind of different damages that a cell may encounter. There are different varieties of damages, and in the first lecture, this lecture, we'll cover pretty much damages that sort of outlined here on the left, right? More so on those on the right, which will be covered in the second lecture. So these are many different types of damages, including alkylating damages, certain events like single-stranded breaks, for example, some lesions that are specifically induced by UV light, just by being exposed to the sun. These form typically genomic DNA adducts. Some errors of replication that was covered early on in the week. For example, these mismatches, deletion or insertions, these are damages that the cells must be able to cope with. And of course, most importantly, a total severing of the DNA, breakage of the double-strand helix. And this is, I cannot emphasize how important this particular type of lesion to the cell viability and the importance of repairing these events with high fidelity. And that will be the entire focus of the second lecture. Additional damages, for example, intra-strand cross-links, these are alkylating adducts that sort of form covalent bonds between the nitrogenous bases in the opposing strands. And obviously, these will interfere with normal transactions in the genome, such as replication and transcription. So these different lesions are then induced by different agents, right? So some agent induces more than one lesion. For example, reactive oxygen species can cause both UV light adducts as well as DNA double-strand breaks. And so one agent can cause more than one lesion. But typically, certain types of environmental insults would cause a particular signature. For example, UV light are probably one of the only sort of lesions that sort of causes these kind of products. So when you see these kind of products in the genome, the 6,4 photo products, for example, or cyclobutane purines, you know that these results from UV fixtures. And so that's a signature for that kind of damage. Some of the same lesions, obviously, are also induced by clinical agents that we use as a therapeutic. For example, as I mentioned, whether it's ionizing radiation or platinum, compounds that are commonly used, or even something like temozolomide, the only agent that is used for brain tumors. All these agents pretty much have the capacity to kill cancer cells primarily through their ability to induce different types of DNA damage. And the more extensive the agent in its ability to induce damage, the more efficacious these agents are. Of course, these agents are not specific. They will target all kinds of cells, and that poses a challenge in the balance between achieving certain therapeutic efficacy versus toxicity to normal tissues. And so that's where the challenge comes from. Of course, the different type of lesions must be repaired, and for that to happen, there are different programs that have evolved to deal with these kind of lesions. And so, for example, in the first lecture, we'll talk about these 4 or 5 different types of DNA damage repair that would deal primarily with some problems that affect the architecture of the genome more so than the integrity of the genome, meaning those that are not necessarily causing double-stranded breaks. And then in the second lecture, I'll talk more about double-strand breaks, which again, the most serious of all, and that's why we dedicated a whole lecture for that topic, as well as programs that deal with repairing cross-linking agents, which also are having the capacity to induce double-strand breaks. Okay, so this is a nice summary slide that you can always refer to back to sort of grasp the main topic of today's lectures. All right, so this is your favorite chemical structure of the DNA. Here at the bottom, just looking at one strand, this is the phosphodiester sugar strand. And then emanating from that are the nitrogenous bases to the inside of the helix. And here are our 4 basic nucleotides, 2 purines, guanine and adenine, and 2 pyrimidines, thymine and cytosine. They are very much similar in structure. Of course, there are some differences. It's not my job to go through these differences, but certainly to highlight the fact that almost every covalent bond in any of these structures can be subject to damage, right? So here I'm highlighting in blue these linkages that are subject to hydrolytic attacks. Just water. All you need is water to induce a breakage of that nitrogen-carbon bond, releasing the nitrogenous base and leaving an abasic site, a site without a base within the helix, right? So if this has happened to be a purine, in this case guanidine, right? You lose the guanidine and you leave an purinic site. If this happened to be in a cytosine and you remove that particular nitrogenous base, then you'll end up with an abasic or pyrimidinic site, right? So that's a challenge, obviously, because now you're removed the coding sequence in the structure of the DNA. So you're only left with the backbone and no genetic information. And so you need to restore that somehow. Another event that can happen to these nitrogenous nitrogen atoms, is a methylation reaction where a methyl group gets to be added in one of these nitrogen events. These are not as predominant as the hydrolytic attack, and so I guess the thickness of these arrows highlight how frequent these events are. You could also have oxidative damage, such as something that could happen to this particular carbon, to this double bond, here, here, and here, as well as in the sugar, in the deoxyribose moiety as well. You could also, of course, have hydrolytic attack in the phosphodiester bonds, so that would ultimately sever the DNA strand if it's removed entirely. All right, so every single base, right, in our genome or in the genome in general, of any organism, can be subject to multiple numbers of modifications that are not normal. Of course, there are modifications that are normal, and you probably will hear about that as you deal with the topic of transcription. For example, 5-methylcytosine, where you add a methyl group to this particular site in the cytosine moiety, and that's just simply become a modified, normal modified base that would suppress transcription. This is a normally inflicted kind of modification and is not necessarily seen by the cell as a damage event. All right, so let's take a look at some of the most frequent spontaneous chemical reactions that can cause some serious damage and how that gets to be produced. So here's again the phosphodiester sugar backbone of one of the helical strands. Again, to the outside is the backbone, and to the inside is the two nitrogenous bases, in this case, guanine and a cytosine. So a depurination reaction is when you basically remove one guanine, right? Or in this case, it's a purination reaction. So a guanine or an adenine, if you remove them, leaving the intact backbone, then that's a depurination reaction. And that requires nothing, as I mentioned, more than just water. You release the guanine, you're left with a naked backbone, if you will, right? You could also, and I didn't mention this here, you could also remove the amine group, for example, by also nothing more than hydrolytic attack in one of these bases, and that's called a deamination reaction. So you obviously release an ammonia, and you're left with a deaminated bases. And so that's the example shown here, where a cytosine, for example, can lose this amino group, and again, that requires nothing but water. You're releasing that, and then what that will leave you with uracil. And guess what? Uracil is not a DNA base, but it's an RNA base, right? So to the DNA, that's a foreign base, right? So it's problematic because it would not base pair normally, and then it causes problem when it gets replicated in the context of normal cellular division. So that also need to be corrected. More examples here, and again, I don't want to be going so much into details about different things, but you can imagine removing an amino group from an adenine that will again release ammonia and will create a hypoxanthine. That's again another unnatural base. Xanthine is another unnatural base. You could have a deamination reaction on cytosine, and that will result in uracil, I just mentioned. You could remove. Remember the example where I just said that not every modification is considered as DNA damage. Here's an example, the 5-methylcytosine. This is a natural modification that can happen to cytosine, particularly at the promoter of certain genes to suppress transcription. But if that amine group gets hydrolyzed, right, then you release ammonia, and that gets changed into thymine. So you're moving from a cytosine to a thymine, and obviously that's a base substitution. And if that's replicated, then you'll end up with a different sequence altogether, depending on where that mutation or that change happened. So how does it deal? How do these mutations then result into a mutation or a problem in the genome? Well, here's an example. So here you have a deaminated cytosine. Again, that's a uracil, right? In the course of normal replication, right, these two strands will separate. The bottom strand obviously have no problem to replicate again, and it will just regenerate the initial or original cytosine, and you end up with an unchanged genomic sequence in one of the daughter cells. In the second daughter cells, however, the uracil that was generated by the deamination reaction will then anneal or will be complemented during DNA replication with an adenine, right? So remember, this was initially a cytosine. And so now the cytosine normally is complemented by guanine, but a uracil will be complemented with an adenine, right? And so that will then result into a U to A substitution, right? I'm sorry, a U to A complementation reaction. And then this strand, if it's replicated again in the next cycle, right, it will create an A to T. And so then you'll end up from a C to G, right, into T A mutation, base substitutions. So that mutation gets fixed in this daughter cells. Now, despite the fact that one daughter cells have the intact sequence, the second one will inherit the mutation. Similarly, if you have a depurinated adenine, an adenine that have lost the adenosine, nitrogenous base, then If this strand separates before you put in the correct bees, this will have nothing to pair with. And so you'll end up with TCT, right? And you ended up with deleting one bees. Of course, the second strand is intact, and that will be replicated just fine. And you'll get, the result will be a cell, a daughter cell that have the same sequence. So a depurination that is not a depurination reaction or a depyrimidination reaction that is unfixed results into point mutation. In this case, a deletion. Of course, if this happened in a cluster of nucleotides, you'll end up with more than one single base deletion. But one single base deletion in a gene, if you remember transcription basics, right, that would result into shifting the frame of coding, meaning that you'll end up with a frame shift mutation that will result into termination of translational activity from that gene. So it can kill the entire function of the gene if that's not repaired. This is how serious a single nucleotide deletion or insertion, or even if you have two. If you have three, it's a little bit different story because now you're maintaining the frame, but you lost one amino acid or so, all right? So the problem really is with frame shift mutations, a deletion that's made of ones or twos of nucleotides, but not threes or sixes or nines or so on and so forth. All right, so here is, now that we've gone over the different type of damage, and these are only some examples and very few examples. I want to overwhelm you with the types of damage that could happen, but these are some of the most commonly seen problems with genomic architecture normally without even some serious damage, and how the cells actually repair that. So the very simplistic way to repair that is to simply repair the damage if possible. And there are some examples of that. In fact, the very first DNA damage repair enzyme was discovered in bacteria belonged to what we call these ALK-B protein. The ALK-B proteins are, again, that was discovered in bacteria. These happen to be an iron-dependent enzymes. What they do is they recognize methylated adenine at position 1. So this is the first position in the adenine purine. And that methylation reaction is obviously seen by the cell as a problem because it's just a foreign base now. It's a methylated base, right? So what this enzyme does is that it uses alpha-ketoglutarate and oxygen. It actually belongs to the what we call the oxyglutarate-dependent dioxygenase family of enzymes. So they're basically demethylases. What their job is to do is to remove the methyl groups. And they do that, right, restoring the adenine through the formation of an intermediate that replaces the methyl group with a formaldehyde, or in this case, a hydroxymethyl group, releasing in the final step formaldehyde reaction, as well as succinate and carbon dioxide. So you can see the enzyme is very efficient at simply removing the lesion. In this case, the damage that just simply gets cleaved off through a two-step enzymatic reaction. Right, so this enzyme is very efficient in doing that. In mammals, it turned out there are actually 9 different homologs of ALK-B, and you can imagine why there are such a high number of different homologs. They're not all identical. They're similar in their ability to utilize iron and utilize alpha-ketoglutarate and oxygen to restore these lesions through direct chemical reversal, but their substrates are different. You know, for example, in this example here, this particular protein uses reverse the alkylating events at position 1. You can imagine another methyl group at this particular one that uses a different enzyme to directly reverse that chemical modification. Okay, so extensive number of homologs, they all belong to the same family to simply remove the damage. Okay, nothing fancy, very simple reaction. Here's another example of this chemical reversal, kind of fancy molecule, actually. It's a suicidal molecule. In this case, it's a molecule that recognizes O6-methylguanine. So here's a methylguanine, right? This is position 1, this is position 6. So at the position 6, you have an oxymethyl group. This is a lesion that can be induced by different alkylating agents. It's a highly toxic, highly mutagenic lesion, and the cells need to repair it. And the way to do that is through utilizing this enzyme. It's called O6-methylguanine DNA methyltransferases, right? So if you just dissect the name of that enzyme, It's an enzyme that recognizes O6-methylguanine, and its job is to methyltransferase, right? It's to transfer the methyl group from the substrate, the damaged bees, onto itself. And it uses that by having a particular cysteine residue in its body that has a sulfhydryl group that can accept that methyl group instead. So what the enzyme does is that it acquires that methyl group away from the lesion, and in the process, it inactivates itself. So it's a suicidal enzyme. Very energetic process. The cell invests a lot of energy to make many, many, many copies of this enzyme in order to keep removing this highly toxic lesion from the cell. Again, another example of direct chemical reversal. All right, so let's take a look at the first sort of programmed way of repairing DNA damage. This is an extensive topic, as I mentioned, and again, my job is not to really highlight every single point in it, but rather to give you an overall description of the different steps that are involved in this particular repair. And from the name, you can see that base excision repair is a repair pathway that excises individual bases. And so the way that these particular enzymes involved in this reaction or in this BER program is that these are a family of enzymes, a family of DNA glycosylases, that they continuously scan the genomic DNA. So what they do is they just simply scan the genome looking for any unnatural beads. And when they find one, they stop and they start undergoing conformational change and acquire new activity in order to remove that lesion, right? So this is something that's been sort of thought of as what we call a base flipping scanning process, where each of the internally faced nitrogenous bases flipped to the outside and scanned through these enzymes to make sure that they are perfectly fine, that there's no damage with them. And when they do recognize these damage, then they stop and they do their job. All right, so this is an ongoing reaction that's happening in the cell all the time. The enzyme is not active until it meets until it recognizes one of those lesions. All right, so its function is to excise damaged bases through their specific activity, and their activity is glycosylation reaction, and there are 6 types available in mammals. They basically remove deaminated cytosine, deaminated adenosine, alkylated oxidized bases, bases with open rings, bases with carbon-carbon single bonds. In the process of repairing the lesion, what they do is they create what we call a basic site, meaning they excise the nitrogenous base, but leaving the backbone intact. And I'll show you this in a second. And so that a basic site is then recognized by an enzyme called an AP endonuclease, and AP stands for apurinic or apyrimidinic. Sorry, there's a mistake here. It's only 2 P's, not 3. Endonucleases that cleave the A basic sites and to generate a 3' hydroxyl and a 5' deoxyribose termini. Adenine polymerase have to come in and then fill the gap by inserting the correct base using the information available from the opposite strand. And then ultimately, the DNA ligase then will come and seal that nick, right? So let's just go over it one more time. So a lesion is recognized by the DNA glycolase during this flipping or base flipping mechanism. They recognize all these different kinds of lesion. There are 6 types of them. They would remove the base. They will make an incision in one of the strand, and that incision will be filled by a polymerase, and then the 2 ends of the separate strand will be ligated together through the activity of DNA ligase. There's only 3 different ligases in mammals, 1, 3, and 4. There's no number 2, but 1, 3, and 4, DNA ligase 1, DNA ligase 3, and DNA ligase 4. We'll go over them in a little bit more detail. But again, I don't expect you to know the individual names of these different molecules. I just want you to really know that there are different 4 steps that they require recognition, they require cleaning up of the bases that are damaged, requiring filling up the gap, and requiring sealing the gap. And that's a theme that you'll see in almost all these different DNA damage repair pathways. Okay, so here's a schematic of that. So here is an example of deaminated cytosine, this uracil that I told you about, that if not fixed, will result into a transversion reaction, converting the originally put cytosine into, this will then complement with the T, putting an A instead at that particular position, causing a T to A transversion. All right. So then this abnormal nucleotide will be recognized by uracil DNA glycosylase. And again, from the name, as it implies, this is a DNA glycosylase that specifically recognizes uracil in the context of DNA. Of course, uracil is a normal base in RNA, but not in DNA. So this enzyme has the capacity to differentiate between DNA molecules and RNA molecules. And when you find one, it triggers the reaction. What it does is then it comes and removes the uracil altogether, leaving an A basic site. An endonuclease will come and will make a severing of that one strand, leaving this base intact. This base then directs the formation of a C by DNA polymerase, and then the gap will be filled by DNA ligase. That's as simple as it is. There are two little versions of that. The first version is what we call the short batch base excision repair. This is what's depicted here on the left. Here's an example of a different lesion, for example, 8-oxoguanine. This is another oxygenated guanine, and requires for its repair an enzyme that's called 8-oxoguanine DNA glycosylase. Right? This enzyme recognizes specifically this lesion, and when it does, it removes that lesion, leaving an A basic site. An AP endonuclease come in, leaving a 3' hydroxyl group and a 5' phosphate at the gap. A DNA polymerase beta, in this case, will come in and fill the gap, along with the function of a bunch of accessory proteins, most important of which is PARP, and recruits the DNA ligase that will then seal the gap and restore the sequence. So again, you start with the lesion on a single base, you remove that damaged base, and you replace it with the correct base using information from the sister strand. A modification of this process is caused by lesions that causes single-stranded breaks, where you actually have breaks instead of a specific lesion. Of course, there's a break here that's a single-stranded break, but this is artificially made in the process of repair. But independent of that, many agents can actually cause these single-stranded breaks. And here's an example of single-stranded breaks that are formed when you have a trapped topoisomerase. Topoisomerases, you probably heard during the DNA replication lecture. These are classes of enzymes that sort of remove the topological strains ahead of a moving replication fork. And the way they do that is that they basically creating a covalent bond at the helical structure. And then they make an incision in one of the strands, allowing for the helix to rotate around the axis, and then it seals it again normally. That's a normal process that happens during replication by topo 1, or during transcription by topo 2, and that just normally get fixed. But sometimes these enzymes get trapped. For example, there are cancer therapeutics actually that are used commonly, topo 1 and topo 2 inhibitors. What they do is that they sort of fix the topoisomerase in a stage where they induce the first event, which is making the single-stranded break, but they do not allow the enzyme to rotate around the axis, and thus creating single-stranded breaks. The way for the cell to overcome these breaks is by recruiting directly a protein called PARP, in this case, poly-ADP ribose polymerase 1. There are different versions of it. But this PARP then recruits a whole bunch of other proteins, including tyrosine deaminated phosphodiesterase, and another enzyme that will be important to sort of clean up the termini at this particular break. What this enzyme does right here, this TDP, is that actually it creates a nick in the DNA, but it has the 3' instead of being hydroxylated, being phosphorylated. So you have a reverse orientation, and these cannot be simply ligated, right? For ligation to happen, you need a 3' hydroxyl and a 5' phosphate group. And so that's where you need an enzyme that will reverse that order. And cells have evolved one such enzyme that's called polynucleotide kinase phosphatase. This is one unique enzyme that has two different opposing catalytic function. One is to add a phosphate and another one to remove a phosphate. And so what it does is that it removes this phosphate, puts a hydroxyl, and puts a phosphate group at this particular position, reversing this orientation and restoring the normal orientation for radioligation in the very final step. But before that has to happen, DNA polymerization has to come and fill the gaps. And because these lesions are much more complicated, cells ensure that you actually remove more than one base and to use the opposite strand to synthesize a longer batch of DNA sequence using polymerase delta or epsilon, along with the accessory factor PCA that you've heard about during the replication lecture, to function as a processivity factor to fill up this gap and restore the entire sequence. This is another way of for the cell to make sure that not only that you're removing the single break, but you also did not leave any modified bases in the vicinity. So it's an efficient way of repairing that. And so because of the longer batch of repair that's been happening here, then this has been termed a long batch base excision repair. Still base excision repair because it utilizes the same factors that were utilized in the subsequent step right here, including ligase 3 and ligase 1. Okay? So now highlighted here in this diagram are a few other features. You'll see, for example, those that are circled here by red, these are enzymes that are actually happen to be upregulated in cancer, right? And so cancer cells are continuously undergoing replication stress. And so cells have evolved a way to actually overcome some of that stress to deal with the ongoing replication problems that these cells encounter. And so because they're overexpressing cancer cells in order to cope with these damages, they became good therapeutic target. And that's why people have actually spent a lot of good effort to trying to develop specific inhibitors that will target these enzymes. And they have been successful. For example, the PARP1 inhibitors, not only for this reason, but for other reasons that we'll be describing in the second lecture, have been some of the most promising therapeutic agents for PARP BRCA1 deficient breast cancers. And there's a reason why they work primarily in this particular context and other types of cancers. We'll go over that again in the second lecture. You also have these other proteins that are depicted here in green, right, circles. And these are proteins that happen to be mutated or silenced in cancer. And so their mutation or their silencing or their deletion, for example, is sort of a way for the cells to actually acquire more mutagenic event. And we know, for example, in the case of cancer, that many of the most aggressive types of tumors, they do have abnormal genome, that their genome is not intact, that they're filled with mutations. And they are filled with mutation, which actually gives them a survival advantage. And new characteristic is because some of these DNA damage repairs have been mutated. And so cells basically can inactivate these genes in order to allow for more room for evolutions of the genomic architecture to give them advantage over normal or otherwise normal cells. Meanwhile, some of these DNA repair molecules, because of the significance of their presence in the cells, such as in the case of PARP, they became an important target that we can take advantage of by simply trying to develop specific inhibitors for which, in order to direct the killing of these cancer cells in particular. We'll talk about that more in the context of double-strand breaks. All right. All right, so that's the base excision repair. Okay, you've got a lesion that's affecting one base, a mismatch, not a mismatch. I'll talk more about mismatch later on, but a particular lesion that's recognized by DNA glycosylases. They come, they remove them, they induce a nick. An endonuclease will do that, and then a DNA polymerase will come in, fill the gap, and a ligase will come and seal the gap. Another type of lesions, such as those that are inflicted by UV radiations, induces what we call bulky adducts, right? So here, what UV light does uniquely, in my opinion, is that they sort of allow for the formation of covalent bonding between neighboring bases on the same strand. And so here's an example of one such conjugation event, or two such conjugation events that affects two nearby pyrimidines, causing what we call cyclobutane thymine dimer. This is one thymine, two thymine, but this has become now a dimer, right? It's a covalently bound as opposed to the normal hydrogen bonding that happened between these bases. And of course, this is very problematic because cells then will have different problem complementing with the opposite strands. Another form of UV-specific lesion, what we call the 6,4 photoproduct. This is covalent bonding between the carbon at position 6 and carbon at position 4 in two neighboring thymine. So these two different forms of lesions will then cause a kink in the major or minor groove of the DNA that can easily be recognized by DNA repair molecules. But these cannot be fixed normally by the simple base excision repair. Now, we have more than one base that has been affected, and it causes a completely different topological problem to the DNA that recognized by a different set of proteins. And that gets fixed through the second mechanism of programmed repair that we collectively call as nucleotide excision repair. Okay, so let's go over this a little bit briefly. You have a problem with a CT dimer, for example, in this case, a pyrimidine dimer, a CT1. An enzyme will come and recognize that lesion. But because these lesions are usually very abundant, what the enzyme does is actually it makes two incisions flanking that lesion on the same strand. So you can see here, it will make one cut here, and it will make one cut here on the same strand. And the idea is to remove the entire batch of problematic strand. Remove it entirely, leaving a gap. That gap obviously must be filled, again, with the DNA polymerase, and then must be sealed with a DNA ligase. Okay, so the only difference really is instead of removing the actual base, you're removing the entire batch of nucleotide, typically 20 to 30 nucleotide long. Okay, so let's take a look at this in much more detail. There are actually two versions of nucleotide excision repair, or NER. The first one is that repair pathway that happens during transcription. And so it's acquired the name of transcription-coupled nucleotide excision repair, or TC-NER. Transcription-coupled meaning that the recognition of that lesion happens during transcription, where RNA polymerase 2 is transcribing that strand. I think you'll cover the topic of transcription maybe in the next block or the block afterwards, so you'll get to learn more about transcription, hopefully much more detail. But for the sake of today's lecture, suffice to say that when a DNA polymerase scans the DNA in order to transcribe that strand, it encounters this lesion and it stops, it stalls. And instead, what it will do, it will recruit two set of proteins called CSA and CSB. They stand for cocaine syndrome A and cocaine syndrome B protein. And obviously the names came from the fact that these two genes, when they have mutations, in other words, individuals that cannot repair that kind of damage, they acquire this disease. It's a genetic disorder that is manifested by the inability to repair lesions that are specifically happening in response to UV light. We'll go a little bit more about this in a second. All right, so in this case, the bulky adduct is recognized in the context of transcription. The other branch is what we call the global genomic nucleotide excision repair. This is the same as the first one, except that it doesn't require RNA polymerase for recognition. This is something that's recognized normally in even non-transcribed genes. So it's a global pathway that can recognize the same type of lesions. But they do not require the RNA polymerase signaling to these two proteins. Instead, they're recognized by these 4 protein complexes that are using two particular proteins called XPC and XPE. And again, I don't expect you to remember them in detail. But suffice to say, all these proteins that are having to acquire XP, this protein, and the subsequent downstream event, XPF, for example, they all belong to what we call the xeroderma pigmentosa group of proteins. These are genes that are involved in this particular process. And when mutated, they give the patients a disease that's called xeroderma pigmentosa. And we'll go over this in a second again. Okay, so in NER, then the lesions are recognized either by RNA polymerase II signaling through cocaine syndrome A or B, or XP factors along with some other accessory factors. From here downward, the process utilizes the same set of proteins, TF2H and XPA, and they will start cleaving the damaged strand far away from the lesion, about 20 nucleotide far apart, and then recruiting additional proteins that includes PCNA along with the DNA polymerase delta or epsilon. These are the normal replicated polymerase. It's a typical replication reaction where the cells uses this intact strand to restore that batch that the cell have removed because it's damaged. And so this is a very fast and efficient process. A DNA ligase 3, the same DNA ligase that seals Okazaki fragment, for example, will come in and fill the gap, restoring the original sequence. All right, so basic excision repair was the first one. We've gone over this. Nucleotide excision repair is more dedicated towards recognizing bulky adducts, particularly those in response to UV light. They are typically extensive in the cells, and therefore the cells have evolved two pathways to repair them, depending on where they're encountered, whether they're transactivating genes or a normal silent genes or gene desert area within the chromosome. They utilizes a different set of proteins to signal to the common downstream elements, ultimately removing the batch that's damaged, filling up the gap with the DNA polymerase along with the processivity factor, PCNA along with RFC, and then ligating the ends with DNA ligase 3. All right, so here are two kids that actually have suffered from xeroderma pigmentosa. And from the name, you can imagine that these individuals have manifest extremely with very highly dry skin, along with some pigmented dark spots in the skin, most commonly in the face, because they're typically results from exposure to UV light. And so this is a disorder that's characterized by extreme sensitivity to sunlight. They cannot relieve that at a very sensitive to these things. They have severe sunburns, freckles problem. They also have telangiectasia, or what we call spider veins. These are all common manifestations of these diseases. Of course, this is not a very common disorder. It's an autosomal recessive genetic disorder that's specifically affecting nucleotide excision repair. So that's a signature for these individuals. And of course, the sensitivity to sunlight is because the sunlight causes these lesions that they cannot repair. So here's a second disorder that affects primarily that branch of nucleotide excision repair that is associated with transcription, the transcription coupled repair. So this is cocaine syndrome. Remember these two proteins that recognize these damage, they're called cocaine syndrome A and B. These are the heterodimer that are signaled to by the RNA polymerase II during transcription. And individuals having problems signaling through this pathway, they develop this cocaine syndrome, or also called as Neal-Dingwall syndrome. A very characteristic feature of this syndrome is the very premature aging features that these individuals encounter. These individuals will have a miserable life. They rarely reach adulthood. They usually die at a very young age. Another examples of that, there's clinical features associated with growth and developmental arrests. They have severe neurological disorders. They are usually having wasting problem. They're extremely sensitive to photosensitivity. Surprisingly, they don't develop skin cancer, maybe because they don't reach more than adulthood. Not because I guess skin cancer in general is a disease of the aging. You know, usually it takes much more than a simple mutation to be manifested as a disease. And so these individuals, like I said, they don't usually live beyond adulthood. Usually die at a very young age. There also, you can take cells from these individuals as well as from XP patients, and you can examine them in culture. You can test them for their sensitivity to sunlight. You can expose these cells and take them from these individuals. And of course, you can take some normal cells from age-matched individual and expose them to different amount of a given wavelength of UV light, and you can measure how much these cells tolerate UV damage. And you can see that, or you will see that cells taken from these patients are easily killed by an amount of DNA damage that's caused by sunlight that would not be harmful at all to normal cells. So you can actually measure their sensitivity to UV light. And this is how these genes were discovered in the first place, by looking at mutations that will make different cells in culture sensitive to sunlight. And then later on, they suspected that such a gene exists in mammals, and they looked for homologs, and they identified these genes, and this is how we discovered them. All right, so these obviously are extremely sensitive to light. They're also hypermutable, meaning that there's a frequency of changing the genomic architecture because of the fact that they're not able to repair these lesions. And of course, there's a problem with RNA synthesis because this is dependent on, unlike the case of XP, they're dependent on transcriptional processing. So if you're not able to repair these lesions, transcription will be halted, and therefore you have problem with transcription, especially after UV exposure when you have more of these lesions available. All right, so that finishes our nucleotide excision repair. So we talked about base excision repair, one damaged base. Nucleotide excision repair, usually more bulky adducts, more severe lesions, maybe clusters of base damages can be corrected by that. And then our third type of damage that is also fixed by programmed repair pathway is what we call the mismatch repair. So mismatch repair, obviously, is a program that is specifically specialized in repairing mismatches, right? Whether these mismatches are different bases opposing each other within the helix, or mismatches because of the presence of an extra or less nucleotide, a deletion or an insertion, that also will be recognized as a mismatch event. Now, one important characteristic of mismatch repair that is very specific to repairing damage during mismatches that originate during DNA replication. And as I mentioned, DNA replication is dependent on the activity of DNA polymerases, and these polymerases are not 100% error-proof. They do form errors, and some of these errors could be in the shape of a mismatch or slippage or the addition of an extra base. They all will be recognized by the mismatch repair apparatus. And again, these happen spontaneously. You don't really have to have an environmental exposure in order to get a mismatch. This is just happening normally during DNA replication. Therefore, this type of repair is much more important in dividing cells than in non-dividing cells, right? Because non-dividing cells do not undergo replication. Once they already reach their maturity and they're dependent on their metabolic activity rather than on dividing to do whatever function they're supposed to be doing, they don't need to replicate anymore. And now they no longer have that capacity to accumulate mismatches during normal DNA replication because it just doesn't happen, right? So mismatch repair then is much more important in dividing cells, and it's much more relevant to cancer cells because cancer cells, by definition, they are losing the control over their ability to hold replication. All right. Clearly, mutation in mismatch repair proteins will result then in extreme sensitivity to alkylating agents because they're not able to fix mismatches that result from these alkylating events. And so let's take a look at that. All right, so mismatch repair, as I mentioned, this is a program that recognizes problems, mismatches during replication, and it has the capacity to recognize the problem in the new strand rather than in the template strand. In other words, the normal parental strand has no problem, but the cells have made or inserted DNA polymerase delta, for example, have inserted the wrong base, right? So the mismatch repair protein recognizes that the problem is not with the parental strand, but rather with the newly synthesized strand. So therefore, mismatch repair is actually strand specific, and that's the beauty of it. Evolution have made its way to make sure that it actually doesn't change the original correct sequence, but specifically recognize the wrong sequence. Right? And that's, of course, is important to maintain the genomic integrity and to maintain the original sequence, especially if that sequence happened to be an important gene. Okay, so what happens when you have that mismatch? When you do have, in this case, is a base pair substitution. For example, an AC as opposed to AT. So the problem with that mismatch is that it's not a normal complementation, and cells have the ability to recognize that. And what recognize that is a set of heterodimer called mismatch repair protein MSH2 and MSH6. This heterodimer would specifically recognize these pair substitutions, and it will then signal to another trimer complex made of two different proteins, MLH1 and MLH3, an accessory factor called PMS2. And again, I don't expect you to remember the names. You'll only become relevant if you really become interested in the damage repair pathways. But if not, you don't have to really remember the name. You just need to know that there are certain proteins that can recognize mismatches in a strand-specific way, right? A different heterodimer that's made of the same subunit MSH2, but now it recruits another subunit altogether called MSH3. And this new heterodimer recognizes insertions or deletions. So here's a strand. Here's the leading strand being made. Here's the lagging strand. So the leading strand is being synthesized continuously, and here's the polymerase came and skipped inserting a base in front of this one. So it actually had a deletion right here. So you're having basically one base on this strand that is not matched with another base on the other strand. So you have a point mutation, and if that's not fixed, this mutation will be inherited as a single point mutation, and that would result into frame shift if it happens to be in a gene, right? Which will then inactivate that gene. So a single event that's not repaired can kill the activity of the entire gene. The entire gene could be 10 kb, 20 kb, 30 kb, right? Nucleotide long. A single event that's not fixed will inactivate the entire gene. That's why it's really important to fix these kind of lesions. And so MSH2 and 3 come together as a heterodimer, recognize these lesions specifically, and signal to the same trimer, MLH1 and 3, and the accessory factor PMS2, and will then signal to this endonuclease 1, which will come and remove the new strand altogether that was just put and start all over. And again, start all over through the activity of the same proteins that are involved in DNA replication, filling up the entire gap and restoring the sequence. Now, no problems. Okay, so it's basically sort of very similar to the proofreading activity of the normal DNA replication. The normal DNA polymerases will have that activity, but if it happens to be bypassed, now you have a backup mechanism that requires activity of these enzymes to actually come and detect these lesions beyond the point where the replication polymerase have passed it and not detected it. And so it's just another layer to make sure that the normal replication have gone correctly. Okay, so basically for normal DNA replication, you have two proofreading mechanisms. The first is the inherent proofreading activity that is associated with the DNA polymerases, delta and epsilon. And second is the mismatch repair pathway that comes in and detects these lesions if they were missed by the proofreading activity of these polymerases. Pretty fancy. All right, so that's what the mismatch repair protein does. Mismatch repair protein also become really important if you have, for example, accumulated alkylating events, such as what we have here, a guanine that, for example, accumulated or acquired a methyl group. So this is an O6 methyl guanine. Remember, this is corrected normally by MGMT that we talked about, this suicidal enzyme that has the capacity of taking up the methyl group and repairing it. So this is part of the direct reversal, direct chemical reversal, the very first step of DNA repair that we talked about. But if you do have a problem with MGM2, either because it's deficient in the cell, because of mutation, what have you, or because you have treated the cells with an inhibitor against that MGM2 transferase, then you've got a problem. Then you've got a fixated methyl group in the genome that will undergo replication. Now, the problem with this is that the problem is not with the new strand. The problem is with the template strand, with the parental strand, right? And if the cell tried to repair that in the absence of the direct reversal mechanism, right, it will actually attempt to repair the newly synthesized strand. The mismatch repair doesn't know, right, which one to repair, except that it's dependent on the fork structure. And in this case, this particular mismatch repair would repair the second opposing new strand, putting in something that will match with the O6 methyl guanine. And that, when it's replicated in the next cycle, will create double-strand breaks and it will be lethal to the cells. So this is what we call the futile mismatch repair cycle, that in the absence of an intact direct reversal mechanism, or in the absence or deficiency of base repair mechanism, right, the cells become dependent on an abnormal way to repair the damage, in this case, mismatch repair, and that's a problematic repair pathway. I mean, we call it cell repair because the cells detect the damage, but it just fixes the wrong strand. And in doing that, actually kills the cell. So why is this important? This is important from not only understanding how problems can arise in the cells, but we can actually take advantage of that in therapy, right? So we know that, for example, today, one of the most effective way of inducing cytotoxicity in cancer cell is by using something like ionizing radiation. That causes a lot of double-strand breaks, also causes single-strand breaks, and so on and so forth, right? But ionizing radiation is not the only mechanism of killing cancer cells. And in fact, for example, brain tumors, they don't respond very well to ionizing radiation. Instead, they respond relatively well to this alkylating agent called temozolomide. This is a DNA alkylating agent that has the capacity of killing the cells, and, but the cells develop resistance to that, right? The way TMZ works is that it induces the G2M arrest. We'll talk about the cell signaling in response to DNA damage maybe in the second lecture. We'll dedicate some slides to that. But for now, just suffice to say that TMZ induces damage, and that forces the cells to halt their DNA replication until the damage is fixed, right? And of course, if the damage is extensive, these cells get killed. And that's how you would hope that the TMZ will function, by killing cancer cells that cannot repair that extensive damage. So what that drug does mostly is that it adds a methyl group at the O6 position of guanine, and so it requires then the activity of MGM2T in the base excision repair pathway or the direct reversal pathway in order to correct these lesions, right? And that's the only actually sort of helpful solution for brain tumors right now. Brain tumors still not manageable disease, and most patients will develop resistance and they'll die. But current effort actually has taken advantage of that by trying to combine TMZ, right, with inhibitors of either BER or direct reversal. So let's see how that works. So TMZ causes O6-methylguanine adducts, right? Alkylating damage, as well as some additional damages. The O6-methylguanine normally is fixed by direct reversal through the MGMT pathway. And if that happens, cells survive, right? But if you have an inhibitor for MGMT, in this case here, there's an inhibitor that was developed that actually promotes the degradation of MGMT. So the way it works through shutting down the expression of the MGMT protein by shunting them towards degradation, so depriving the cells of MGMT protein. So an example of that drug will be an O6-benzylguanine. This is a drug that will then degrade the MGM2, and if you don't have MGM2, cells depend on repairing these guys through the mismatch repair pathway, and in its absence, in the absence of MGM2, cells will become sensitized to this lesion. Of course, if this is intact, cell survival will proceed, and mismatch repair will be just fine. The second lesions are dependent much more on the base excision repair. These lesions are requiring the base excision repair pathway for repair. And here, if you add an inhibitor for the AP endonuclease, which is an important component of the BER pathway, then you're also depriving the cells from the capacity to repair these lesions. And so then you're ending up with more cytotoxicity. So this idea of synthetic lethality, right, by on one hand, increasing the amount of damage to the cells, and on the other, depriving the cells from a normal repair pathway, right? Because you know that these cancer cells are dependent entirely on that pathway. So if you target that particular pathway, then you have a window of killing these cancer cells with much lesser amount of DNA damage. So that's a very important concept in cancer therapy today. Many examples of that. Okay, so in the last 5 minutes, what I will do is I will try to go over this other mechanism that is not necessarily a DNA repair program, but it's a way for the cells to cope with excessive amount of damage that it's suddenly acquired. And so this is a mechanism of tolerance more so than repair. So even though we classify it among the DNA repair pathways, it's really a tolerance pathway. But obviously, because it's a tolerance and it's not really repairing the damage, this pathway is associated with a high rate of mutations. So let's see what are the concepts behind this particular pathway. So we know that the cells have different types of polymerases. For example, we know that the normal DNA polymerase alpha and the alpha, delta, and epsilon, they belong to the DNA replication, which are basically having a very high fidelity rate. They only make one error in every million or more event. In other words, for every million base pair, they only make one error. And then they go back and they proofread that. Mismatch repair enzyme, you can imagine, the one that we just talked about, there are very high fidelity enzymes. They basically do not make errors. They remove always the mismatch repair protein, and they replace that with the intact sequences. But there's a second classes of, or a third classes of DNA polymerases that have a very low fidelity, but very high processivity. In other words, they don't care very much for what is the base in front of them in order to insert the opposite base. So they don't care very much about the complementary strand. All they need to do is to just processively insert nucleotide. So these are what we call the Y family polymerases, and they are very low fidelity polymerases. And the reason why they're low fidelity is because their catalytic cleft is much more expanded compared to the catalytic cleft of the, so the high fidelity polymerases have a very tight catalytic cleft that it can easily and nicely, with very high specificity, recognize the base against which they need to polymerize the DNA. Whereas the low fidelity enzyme, they had a much wider cleft that it can accommodate any other base. And so the process of tolerance here then is to allow for the cells that when it encounters many of these DNA problem is to quickly fix these lesions, because the cells realizes that it cannot, if it's going to be trying to repair every single site, is just not going to have the time to do that. And so this is a much more efficient way to just skip the lesion, skip the lesion, skip the lesion, no matter what the base that you're going to be putting in will be. And that's why it's highly mutagenic. But for that to happen, the cell utilizes what we call the switching mechanism, where it's dependent entirely on this protein trimer PCNA that we talked about during DNA replication. Again, this is a processivity factor, and it's necessary for the processivity of all polymerases. So normally, it's during normal DNA replication, it's associated with polymerase epsilon, for example, delta, alpha. These are the three different polymerases associated with replication. But when it encounters a significant amount of lesion, what happened then is that the PCNA will switch, will kick out the normal polymerase, and will then bind to another polymerase that is much more processive, but also now compromised with its fidelity. And so it switches polymerase eta, for example, in this case, versus polymerase epsilon. There are many other TLS polymerases in addition to eta. There's zeta, eta, kappa, and so on and so forth. They all have the same issues. They are low fidelity polymerases. And so when they switch, then because these enzymes can't tolerate the lesion, they just go on and replicate the damaged DNA, leaving the damage intact. And of course, in the process, they may insert the wrong base, and then it switches back to the normal epsilon until it encounters the next lesion, and so on and so forth. So one key step in this TLS for it to happen is that it's dependent on a particular modification that happened to the PCNA trimer. And that modification is a process that's dependent on conjugating the PCNA with a single molecule of a small moiety that we call ubiquitin. I don't know if you heard about it or not, but if not, you will for sure by listening to maybe some of the stuff that researchers like me or somebody else in our department that would be talking about protein ubiquitination. So ubiquitin is a very small protein pretty much unstructured, about 7 kilodalton in size, that can be conjugated in a poly chain fashion to different proteins, typically shunting them for degradation. Some proteins like PCNA can acquire one single ubiquitin moiety on a particular lysine residue that gets signaled to in response to this recognition. So when PCNA encountered these lesions, two enzymes come together, RAD6 and RAD18, and the function of these two enzymes is to put in one ubiquitin moiety on PCNA. Okay, so these are what we call E1 and E2 ubiquitin conjugating enzyme that will come together, add one ubiquitin to PCNA. So now this one ubiquitinated PCNA is different than the normal PCNA. It doesn't have high affinity for polymerase delta, epsilon, or alpha. Instead, they now have, or the monoubiquitinated, mono meaning one, monoubiquitinated PCNA now have a very high affinity for these Y family polymerases. And so this is where the switching happened. So the switching is dependent then on the modification of PCNA through ubiquitin conjugation that allow them to lose affinity for the replicative polymerases and acquire affinity for the translation DNA polymerases. Okay, so that's an important signal for that pathway to be kicked on. Okay, so that sort of marks the end of lecture 1. And what I'm having in this slide is a summary of the major programs, skipping the very first step, which is the direct reversal, because it's, even though it's utilized, it's very simple in nature. But those that are really highly programmed include the base excision repair. And as I mentioned, I'll try to go over it so that you hopefully take that as the main message of today's first lecture, is that this particular repair pathway removes one damaged base, right? Typically, these damages come in the form of oxidation, alkylation, or deamination. And this is the one pathway that removes the majority of DNA damage that happen in the cells, whether they're spontaneous or inflicted by certain environmental agents, such as alkylating agents. The second branch is the longer form of repair pathway, the nucleotide excision repair. We talked about two subclasses, one that is associated with transcription, we call it transcription coupled nucleotide excision repair, and one that is globally impacted, global genome nucleotide excision repair. The first one depends on two proteins called cocaine syndrome A and B proteins. The second one depends on a class of molecules collectively known as xeroderma pigmentosa proteins. Then they're processed similarly afterwards, and they have the capacity to recognize thymine dimers and bulky adducts. And typically, mutation in the enzymes in this branch result either into cocaine syndrome that we heard about, or in xeroderma pigmentosa that we've seen some examples of. Then the third branch is the mismatch repair, and we said that this is a replication-specific repair program, meaning that it's a program that has the capacity to function as a backup to problems that initiate during replication, particularly mismatch problems, and that same program can take off also in the case of deficiency of the base excision repair, and people are now trying to take advantage of the capacity of the cells to erroneously correct these lesions to induce more damage in these cancer cells that may have some deficiency. And then we talked finally about this other repair, or more appropriately called tolerance mechanism, where the damage is simply very extensive and the cells basically loosen their grasp on the making sure that the repair is correctly damaged, but correctly repaired, but rather, okay, let's just seal the gaps, let's just continue replication in the face of this extensive damage. And that requires a switching event of the PCNA molecule from the high fidelity polymerases towards the low fidelity polymerases through a reaction that requires the modification of PCNA through ubiquitin conjugation. Okay, so with that, I think I stop here, and we'll see you in the second lecture when we talk about double-strand break repair. Okay, goodbye.